So these are the constituents of different lipoproteins. That is, chylomicrons is made up of 90% Tg, while VLDL is made up of only 60% Tg. LDL constitutes about 8% Tg, while HDL is having only 5% Tg. Now see the apoprotein. Exact opposite, right? So chylomicron is having the least quantity of apoproteins, followed by VLDL. And as we go down the chart, we can see that apoprotein concentration increases. Okay, so as apoprotein concentration increases, what happens? The density also increases, and that is making more healthier, right? So these are the different classes of lipoproteins. Now, what are the functions of lipoproteins? Before that, this is again another cross section, a schematic representation. So these yellow colored substances, these are the polar head of the phospholipids, right? There are cholesterol. These are the free cholesterol molecule, which is seen in the dark blue, right? And we are having the integral proteins. These are the integral proteins, and surface proteins can also be there. Surface apoprotein and surface integral apoproteins. And these are the cholesterol esters, while these are the TGs. Okay. So these we have seen that as apoprotein concentration content increases, what happens? The density also increases. Apoprotein concentration increases, the density also increases. So this is a chylomicron. See, the size of the chylomicron is the largest. Even though it's having the largest size, the size is made up of, or the size is due to the lipid content, not due to the protein content. So it is having the maximum TG and cholesterol esters incorporated within it. And it is having different types of apoproteins like ApoB48, ApoC2, ApoA1, and ApoE. These are the different types of apolipoproteins present on the chylomicrons, right? Either A, C, E, etc. What integral proteins are? Surface protein. It is a surface protein or peripheral proteins, right? Apo A, C, and E. While B48 is an integral protein, right? Now this is the VLDL. See, the number of quantity of TG and cholesterol esters is reduced, so the density is increased, and it is made up of B100, ApoB100, right? VLDL is made up of ApoB100, and this also is having ApoE and ApoC2, right? And the next is the IDL. IDL is having again reduced quantity of, or the content is reduced, so the size is also reduced. It is having ApoE and ApoB100 as its apoproteins. And next is the LDL. LDL, the function of LDL is to transport cholesterol esters from the liver to the extrahepatic tissues. And it is the only lipoprotein having B100. It's having only B100 in its structure. So the LDL contains only a single apoprotein, which is the ApoB100. Now next is the HDL molecule. Again, the cholesterol, the TG is least transported by HDL, and cholesterol esters is also reduced, but again it is transporting the cholesterol esters from the peripheral tissues to the liver, right? So these two, LDL and HDL, are transporting what? Cholesterol esters, while all the other remaining lipoproteins transports both TG and cholesterol or cholesterol esters, while LDL and HDL transports mainly the cholesterol esters. The TG content is very less when compared to that of the other chylomicrons. And HDL also transports, of course, phospholipids are there. Then ApoA1, E, and C2. These are the apoproteins present on the HDL, right? So that is the apoproteins. Now that is based on centrifugation, based on the sedimentation or the flotation constant. Based on the flotation constant, the chylomicrons forms the topmost layer, followed by VLDL, IDL, LDL, and HDL. Now, second mode of separation is by electrophoresis. That is when a power is applied due to the potential difference. Charged species starts moving from the cathode to the anode, and as the protein content increases, what happens? The speed or the rate of mobility also increases, and hence HDL moves the fastest, followed by LDL, then IDL, then VLDL, and the least moving lipoprotein is the chylomicrons. Okay. Then based on the nature of the apoprotein content that we have already discussed. Now, what are the functions of apoproteins, and what what is the significance of apoproteins on lipids? The proteins present on lipoproteins are known as apolipoproteins, and they have got many functions to be carried out. The main function is to transport the lipids from one area of the body to another area, right? Thereby providing stability for the lipids. Then they can also act as ligands for interaction. Ligand means they enables the lipids to get bound to another surface, right? So lipids are hydrophobic in nature, so they cannot interact with other areas or other cells. So this interaction, cell to cell interaction of the lipids is made possible with the help of apolipoproteins or apoproteins. Our apoproteins acts as the binding agent for the lipids. We call it as ligands. Then they are also acting as cofactors for various reactions, chemical reactions in lipoprotein metabolism. Then there are two types of apoproteins: integral proteins, ApoA and B, and peripheral proteins, ApoC and E. A to a certain extent can also act as peripheral protein, but mainly the integral proteins classify A and B are integral proteins, and C and E are peripheral proteins. Now the site of synthesis. I have told you ApoB48 is synthesized in the intestine. It is the only ApoB48 is synthesized only by intestine. In all the other cases, liver is acting as a synthesizing site, but ApoB48 is synthesized individually by intestine. Or intestine is the only site where ApoB48 is synthesized. അതുകൊണ്ട് തന്നെ chylomicrons are made up of ApoB48. ഇത് പഠിക്കാൻ എളുപ്പമാണ് കണ്ടോ ഇവിടെ മാത്രമേ B48 ഉള്ളൂ. വേറെ ഒന്നിലും I haven't mentioned any B48 because B48 is seen only on chylomicrons because it is synthesized only in the intestine, and the function of chylomicron is to transport the TG from dietary TG from the intestine to the liver. So B48 actually B48 പറയാൻ കാരണം B48 is actually synthesized from ApoB100. ApoB100 in the genetic level mRNA splicing എന്നുള്ള ഒരു phenomenon. So gene cut ചെയ്തിട്ടാണ്. When if you want to reduce the size or length of your hair, what will you do? You'll cut the hair, right? So similarly, gene rearrangement ഒക്കെ സംഭവിക്കുമ്പോൾ splicing, the cutting is known as splicing. So mRNA splicing will be happening. That splicing of B100 leads to synthesis of B48. So B100 is actually the parent protein, and from the B100, B48 is synthesized. So B100 moves into the intestine, and there the splicing happens to give rise to B48. So mRNA splicing നടക്കുന്ന സ്ഥലം, it is only the intestine. That is why B48 can be seen only in the intestine or in the chylomicrons. Right. So ApoA1, A2, A5 are there. There are three types of ApoAs. While all the proteins are synthesized by liver. ഏത് apoprotein എടുത്താലും ലിവറിൽ ഉണ്ടാവും except B48. Okay, and intestine synthesized B48 of course, and apart from that ApoA1 is also synthesized by the intestine. And what is the function of ApoA1? It is present on HDL. Then നമ്മൾ നേരത്തെ പറഞ്ഞ functions they can act as cofactors for enzymes, right? So ApoA1 is a cofactor, right, for the enzyme LCAT, lecithin cholesterol acyl transferase. Lecithin cholesterol acyl transferase, an enzyme coming in metabolism. LDL metabolism, cholesterol metabolism varana or enzyme ആണ് LCAT. So LCAT എന്നുള്ള enzyme function ചെയ്യണമെങ്കിൽ it require ApoA1 as a cofactor. Then ABCA1 ligand. You don't have to make by heart these functions. Just go through it, right? Important guideline I have written in the notes. You just go through it. ApoA2 inhibits ApoE binding to the receptors. ApoA5 it activates lipoprotein lipase. ApoA5 the apoprotein activates lipoprotein lipase while inhibits VLDL and TAG. ApoB100 it is a structural protein and LDL receptor ligand. B48 it's a structural protein of chylomicrons. Then ApoC1 it's an LCAT activator. It's an activator of LCAT, right? Then ApoC2 is LPL cofactor. What is LPL? Lipoprotein lipase. Then ApoC3 it's R binding remnant. ApoE ApoE is found on liver, brain, small intestine, gut, etc. It's an LDL ligand. Then ApoA. Yeah. What is ApoA? It is to be taught separately. See, ബാക്കി എല്ലാം ഞാൻ capital letters ലാണ് എഴുതിയിരിക്കുന്നത്. പക്ഷേ this one, Apo, this is known as apolipoprotein A എന്നാണ് ഇതിന്റെ പേര്. Apolipoprotein A. See, this is written in small letters because this apolipoprotein, it's a lipoprotein, apolipoprotein A, it is otherwise known as little rascal. It is known as little rascal. Why it is known as little rascal? Rascal എന്ന് പറഞ്ഞാൽ എന്താ വികൃതി അല്ലെ, അല്ലെങ്കിൽ കുറച്ച് നോട്ടി ആയിട്ടുള്ള, right, ആയിട്ടുള്ള ആളെയാണ് we called as rascal. And the same thing is carried out. വളരെ disturbing ആണ്, വളരെ annoyance ആണ്. Why? Because this, if lipoprotein, this apolipoprotein A is present in high quantities, it is responsible for clot formation. ബ്ലഡിന് ക്ലോട്ട് ചെയ്യിക്കാനായിട്ട് ടെൻഡൻസി കൊടുക്കുന്ന ലിപ്പോപ്രോട്ടീൻ ആണ് അപ്പോലിപ്പോപ്രോട്ടീൻ എ. ക്ലിയർ? സി, ഈ എ ആണ്, മറ്റേ എ അല്ല. ദിസ് ഇസ് അപ്പോ. ഇത് നല്ലതാണ്, പക്ഷെ ഇത് കാരണം ഇത് HDL ലുള്ള കോൺസ്റ്റുവന്റ് ആണ് ApoA1. ApoA1 HDL is a good cholesterol. But ഈ ApoA1 should never be misunderstood with ApoA. This is bad. It is not at all good because it is responsible for clot formation, cardiac arrest, heart attack ഒക്കെ ഉണ്ടാക്കാൻ മെയിൻ കാരണം due to the high concentration of lipoprotein A. കാരണം this LPA, lipoprotein A, is having a close structural similarity with the protein plasminogen. We know what is plasminogen. Plasminogen എന്താ ചെയ്യാം? ഫൈബ്രിനോലൈസിസിനെ എൻഹാൻസ് ചെയ്യും. പക്ഷേ what does apolipoprotein A, the LPA, what does do? It is having close similarity. Appo plasminogen vandirikkunna sthanathu LPA vandirunnitte, it inhibits the fibrinolysis so that clot will be found. Clear? So that is a that I'll show you the this is lipoprotein A, LPA. It is composed of an LDL particle and has a second apoprotein, ApoB100, right? That is LDL B. It is LDL contains only ApoB100. If LDL in the LDL transports the cholesterol esters, right? LDL transport cholesterol esters from the liver to the peripheral tissues, and hence it is having only ApoB100 only on its surface. If an LDL molecule is having cholesterol esters packed in it and having an additional apoprotein than ApoB100, ApoB100 kude, if it's having another apoprotein like ApoA, which is attached to ApoB100, then we call that lipoprotein as lipoprotein A. It is small letter A, not capital letter. Tettana. And a small letter A. Clear? So it is not responsible for binding the lipids. Lipid binding function. It is having this ApoA is having a close similarity to plasminogen, and it interferes with fibrinolysis of thrombi and on the surface of plaques. That is plaque formation enhancing. The plaque is nothing but atherosclerosis. Heart cardiac arrest and the blood vessels in the cross area കുറയ്ക്കുക. എന്തുകൊണ്ടാ? ഫാറ്റിനെ ഡെപ്പോസിറ്റ് ചെയ്യാം. ബ്ലഡ് ഫാറ്റ് വന്ന് ഇങ്ങനെ ഡെപ്പോസിറ്റ് ചെയ്യും ബ്ലഡ് കാപ്പിലറി വാൾസിൽ. അപ്പൊ ബ്ലഡ് ഫ്ലോ അവിടെ ബ്ലോക്ക് വരും. ക്ലിയർ? ആ ബ്ലോക്ക് ഉണ്ടാക്കാൻ എൻഹാൻസ് ചെയ്യുന്ന ഒരു ഫാക്ടർ ആണ് വാട്ട്? ApoA. Because ApoA is having plasminogen, having a close similarity with the plasminogen. Plasminogen വന്ന് ബൈൻഡ് ചെയ്യേണ്ട സ്ഥാനത്ത് ApoA വന്നിട്ട് അവിടെ ബൈൻഡ് ചെയ്യും and induces atherogenesis. Fibrin, what do you mean by fibrin? Fibrin is a clot. Fibrinolysis clot fibrinolysis block thrombus formation form. That is atherosclerosis. It induces atherosclerosis. That's why it is known as a little rascal. It is known as little rascal. Okay, so that is the lipoproteins, right? And functions, it provides recognized sites for cell surface receptors, act as coenzymes. They are acting as ligands. They form the structure of lipoproteins, act as cofactors. They act as receptors on the tissues. Example, ApoB100, ApoE, etc. I've given functions in the notes. Okay, next we'll start with the metabolism.